This is a continuation of Chapter 10. This is Part 3 of Sexual Reproduction and Genetics. In this section, we will take a look at how Mendel solves the monohybrid Punnett squares. So we left off talking about the genotype, which is the actual genetic makeup of an organism. It's what you are. And the phenotype, which is the observable characteristics, or it's what you look like. Now, Mendel knew that taking one pair of contrasting traits was a good place to start for his experiments. So he performed what we called monohybrid crosses. Mono, of course, meaning one. So Punnett squares are used to predict probabilities of the traits that offspring receive. It's a good paper and pencil tool. You can do Punnett squares without actually having to manipulate fertilization of any kind for any organism. So we'll go over the three types of monohybrid problems that you will encounter. We'll take a look first at probably the easiest example, taking a look at crossing two homozygous individuals together. Remember, homo means the same in Latin. So we'll kind of copy what Mendel did for his example. We'll take a look at pod color. There are two colors for this. Yellow is dominant and green is recessive. And for each one of your monohybrid practice problems, you will be given this information. So we'll take a homozygous yellow pea and cross that with a homozygous green pea. Okay, the genotype, that's our two capital Y's. Yellow is that dominant trait, so if it's homozygous dominant, that means we have two capital letters. The homozygous green pea plant, the genotype is lowercase y, lowercase y. Remember, we're talking about one allele with two different forms, so we have to use the same letter. So the genotype, lowercase, lowercase, that's how we know it's recessive. And the phenotype then is it will appear a green color. So in a monohybrid Punnett square, it kind of looks like a window frame. You're going to place one parent's alleles on the left side of the Punnett square and the other parent alleles on the top. One allele per box in a monohybrid problem. It doesn't matter which parent you put on the left and which parent you put on the top. You will get the same results inside the box. So you'll fill in the boxes by combining the alleles. Remember, the capital letter comes first. So in each box, we have one capital and one lowercase. And inside the box, the dominant allele will be placed first. So the result of this is we have 4 hybrid pea plants, and they all appear to be yellow. So the genotypes are 4 heterozygous or hybrids with the capital Y, lowercase y, and we will have 4 yellow plants for the genotype. Now let's take a look at if we cross a purebred individual with a hybrid individual. So in this case, for the heterozygous individual, the alleles will be different. We'll have a capital and a lowercase. So let's take a look at rabbit coat color. And I will be completely honest, I make these things up. I do not know if this is the actual case. It could be, for all I know, but we'll just pretend. So rabbit coat color, we will say that black fur is dominant to brown fur, which will be recessive. So we will take a homozygous dominant black bunny rabbit. Homozygous means the alleles are going to be the same size. And we said it was dominant, and it's a black color, so we'll use the letter B. Dominant, homozygous, big B, big B. The heterozygous individual, remember black is dominant, so it will have the phenotype of black fur, but if it's heterozygous, you will have one capital and one lowercase b. So again, we're going to put the parents along the left side and on the top. Combine the alleles together in the box. Capital letters go first. So we will have, in this case, for genotypes, we will have 2 purebred and 2 hybrid individuals. However, they will all have the same phenotype. They will all have black fur because black fur is dominant over brown fur. Let's take a look at one more example. We'll take a look at what happens when we put 2 hybrid individuals or 2 heterozygous individuals together. Now this I know for a fact is an actual trait. There's a certain type of goldfish that's called a Moor goldfish. Moor is coming from a black color. The black color is dominant, and the recessive of this allele is what they call panda. So we will take 2 heterozygous Moor fish and cross them together. So if we're going to use capital M, if a heterozygous individual is had, we will have capital M, lowercase m. So again, we put our parents along the left side and along the top. Combine what you see inside the box. Capital letters go first inside the box. So here for genotypes, we have still a 50-50 ratio of purebreds to hybrids. Our 2 purebreds would be the capital M and the lowercase m because they are homozygous. Then we will have 2 hybrid individuals or heterozygous with the capital and a lowercase m. Here's where we get that 3-to-1 ratio, that 3 dominant to 1 recessive ratio for phenotypes. So we will have 3 black Moor fish, and we will have 1 panda Moor fish. This concludes part 3 of chapter 10 on sexual reproduction and genetics.